In this presentation, I'm going to discuss whether metformin is deserving of its name as a miracle drug, because that's what a lot of people call it. Let's begin with the background. It's best known for treating type 2 diabetes. It's a standard drug that a lot of doctors prescribe. It is a galagine derivative, so it was made from galagine, and in the medieval times, people used galagine as an herb. However, we now know that it itself is too toxic, so instead we use metformin, which is a derivative. Unlike modern drugs, metformin wasn't designed to target a particular pathway, and this is why we still don't know what the mechanism of action is. We don't know what it does in the body, all we know that it works. However, we do have a few guesses, and the guesses are pretty well studied, and I'm going to discuss it later in this presentation. Despite being a standard type 2 diabetes drug, other benefits have been found as well, and in my opinion, the most fascinating effect is on aging. So let's begin with the formulation. It is made with a hydrochloride salt because salts enhance absorption. This is true for all drugs. Drugs aren't usually in their pure form, they're mixed with something else. So these are the four types of formulations. The first one is modified release. It spreads absorption metformin along the gut. It reduces local concentration, and it's aimed at increasing tolerability. So in this formulation, they don't want all the metformin to clump up in one spot. They want to spread it along the gut. The second is delayed release, DR. It targets the ileum, which is the part of the small intestine, and it achieves this by dissolving at a particular pH. The ileum is at a particular pH, and chemists can design it so that the tablet, dissolves there exactly. The third one is immediate release. So as the name suggests, it immediately dissolves. And the last one, extend release, XR, has lesser side effects than IR metformin. And below is a picture of XR. And above is the formulation. It's made with the hydrochloride salt. So how does metformin work? Metformin uptake is through OCT1, which stands for organic cation transporter. And metformin itself is an organic cation, so it makes sense that it goes through the organic cation transporter. Second, it's absorbed from the small intestine, 70% of it, and the remainder is excreted in the urine. And it's also highly concentrated in other organs such as the liver, kidneys, and bladder. Something to note is that it accumulates in the mitochondrial cells 1,000 times more. This is because in the electron transport chain, protons are pumped into the outer membrane, which makes the inner membrane and inside the mitochondria negatively charged in relation to the outer membrane. This is good for metformin because since it's positively charged and the inside of mitochondria is negatively charged, it's really good. And it can move freely within the mitochondria. Another thing to note is that these are the possible pathways, and in this presentation, we're going to focus on the ones relating to the mitochondria, particularly MGPD and complex 1. Let's talk about the inhibition of complex 1. So the inhibition of complex 1 prevents the production of ATP. This is because complex 1 is a transmembrane protein within the ETC, and electrons can't flow through it if it's inhibited, and H+ ions can't get pumped, so there's no gradient. There's an inverse relationship between ATP and ADP because during cell respiration, ADP is turned into ATP, and most of it is generated in the ETC for oxidative phosphorylation. And there's also a relationship with AMP because adenylate kinase reaction turns ADP into ATP and AMP. Therefore, if there's a lot of ADP, that means there must be a lot of AMP. Though this reaction produces ATP, it's much less compared to oxidative phosphorylation since this is only substrate level. And it's also good to note that AMP activates AMPK. The AMP from the decrease in ATP and increase in ADP and the increase of AMP activates AMPK. The AMPK function is that it phosphorylates ACC1, ACC2. That just enhances insulin sensitivity, which makes your cells use insulin more efficiently. Second, it phosphorylates PDE4B and lowers CAMP. And lastly, it phosphorylates CRTC2, which we're going to discuss in later slides. Let's talk about the gluconeogenesis and glycolysis pathway. So gluconeogenesis is the formation of glucose from non-carbs, and glycolysis is the breaking down of glucose. So in this pathway, when there's high CMP, which means there's low glucose, pKa is activated, which switches off glycolysis and switches on gluconeogenesis. This makes sense because if you have low glucose in your bloodstream, you're going to want to produce more. If you have a lot of glucose in your bloodstream, there's going to be less CMP, which switches on glycolysis and switches off gluconeogenesis because you don't need the extra glucose. You want to break it down and turn to energy. It does this by the inactivation of PFKFB1. The inactivation of PFKFB1 decreases F26BP, which inhibits FBPase. FBPase is a gluconeogenesis inhibitor, which means that a decrease in F26BP switches on gluconeogenesis. So pKa also phosphorylates transcription factors, increasing the transcription of these two genes which aid in gluconeogenesis. This does this because when transcription factors bind to the promoter, it can recruit an RNA polymerase to the DNA, which transcribes these two genes, which are then translated into proteins that aid in gluconeogenesis. And the drug, metformin, it actually lowers CAMP through the PDE4B, which switches on glycolysis and switches off gluconeogenesis. Because if someone has diabetes, you want to get rid of glucose in their bloodstream. This is why you switch on glycolysis. You want to get rid of it as fast as possible. You don't want to produce more. CRTC2 also plays an important role. So the phosphorylation of CRTC2 counteracts the effect of pKa on PPCK and G6Pase. And those are enzymes that aid in gluconeogenesis. So an increase in pKa creates more gluconeogenesis proteins. So the phosphorylation of CRTC2 makes it stay in the cytoplasm, so it cannot act, and therefore PPCK and G6Pase will have less of an effect. Another pathway is the glycerol phosphate shuttle. So the function is to regenerate NAD+ from NADH because NAD+ is a very important molecule. It's necessary to convert lactate into pyruvate into glucose in gluconeogenesis, so it's responsible for the production of glucose. So clinically speaking, glucose production was inhibited after 1 hour of IV administration of metformin. And the lab people, the doctors, they discovered that it associated with an increase in lactate pyruvate ratio, which suggests that there's a problem with the glycerol phosphate shuttle. So in the image below, if you take out MGPD, you get an increase in G3P, which causes a decrease in NADH and an increase in lactate. Since lactate can't be converted into pyruvate into glucose, there are low levels of glucose. So metformin, by inhibiting MGPD, it indirectly affects glucose concentrations by focusing at the lactate pyruvate ratio. The more interesting aspect of metformin is its effect on aging. Some argue that aging itself is a disease. It's been recognized by the World Health Organization. But fundamentally, cells and gene regulation are less efficient with age. As we get older, we become less efficient. However, no methods have been discovered yet to reverse aging, but there are methods to slow it, and metformin is one of them. And recently, metformin gained FDA approval for clinical anti-aging trials. So, how does metformin slow aging? The short answer is that we don't know. We don't even know how it works as an anti-diabetic drug, and this pathway is a lot more complex. However, we're going to discuss some interesting hypotheses. The first hypothesis is the mitochondria, and mitochondria is a focus point for aging because the mitochondria declines over time. So the first possibility is due to mitochondrial DNA mutations. So mitochondria has its own DNA because a long time ago it was its own prokaryotic organism. And every mitochondria, there are 2 to 10 copies of the mitochondrial genome. Second is nuclear genomic instability, which means that the DNA becomes less stable and there are a lot of mutations in the DNA over time, which is bad. Third is reduced mitochondrial biogenesis, which mitochondrial biogenesis is more mitochondria in the cell. So reduced mitochondrial biogenesis means that there's less mitochondria in the cell, which means less energy is being produced. Fourth is defective mitophagy, which is degradation of damaged mitochondria. So mitochondria, they don't have an infinite lifespan. So when a mitochondria isn't working, it's necessary to get rid of it and degrade it. However, over time, you become less efficient and your body isn't as good as getting rid of the damaged mitochondria. The last one is the altered mitochondrial dynamics and ETC regulation. So that means it just becomes less efficient in producing energy. So combining all five of them, your mitochondria in your cells will become weaker over time because of those five reasons. And mitochondrial decline is directly attributed to aging, and it's inevitable. However, metformin has a way to target it. So it does this by regulating the production of reactive oxygen species, which is ROS. So ROS is good and bad. Too much of it can damage your DNA and cellular processes. In the image, you can see that it can do DNA damage, enzyme degradation. So overall, too much ROS is really bad for you. ROS is produced by the mitochondria, and it happens a lot in cell respiration. So mitochondria, by targeting mitochondria, metformin can regulate the ROS in the cell. So by reducing ROS, it does this by reversing electron flow in complex 1. We discussed earlier that it inhibits complex 1, that metformin is a complex 1 inhibitor. However, when it reverses electron flow in complex 1, it reduces the ROS. By elevating ROS, it can cause transcription of SKN1, which promotes longevity. So ROS isn't entirely bad because good levels, controlled levels of ROS, transcribes a gene that promotes longevity, so it's good for you. And SKN1 is directly activated by ROS, so we need ROS in the cells, but too much is really bad. The next thing is telomeres. So by definition, these telomeres are tandem repeat sequences at the end of chromosomes. So what that means is short segments of nucleotides are being repeated and they're placed right next to each other since tandem repeat. It doesn't code for anything, so it's non-coding, so they're not transcribed into proteins. These are necessary because it increases stability of chromosomes, and more importantly, it solves the end replication problem. So after every round of division, the DNA length decreases. So during DNA replication, it's semi-conservative, so there's leading and lagging strand. This is because DNA polymerase can only add new nucleotides if there's a 3' OH. So therefore on the lagging strand, you must place continuous primers and Okazaki fragments, and you need to ligate them together. So at the end of the lagging strand, you're going to have an RNA primer with DNA on it. However, you can't use an RNA primer in DNA, so the RNA primer must be removed. However, you can't fill in that gap because there's no 3' OH that polymerase can add to, so you're just missing that chunk of DNA. And over time, that can add up, which is a really big problem. And cells combat this by having telomeres. By getting rid of this junk code, which is in the telomeres, you can prevent the actual good coding DNA or non-coding DNA from getting removed. However, telomeres aren't infinitely long and they can shorten. So when telomeres are too short, the cell stops dividing and it senesces. So it just stops dividing. However, some cells, especially cancer cells, they can have telomerase, which makes it live forever, which is a bad thing. However, most of our cells, we don't have telomerase, and our telomeres can get short over time. So telomere length can be thought as a biological clock. So metformin targets telomeres by reducing telomere shortening. So if you reduce the telomere shortening, then therefore they can last longer and your cells will take more time to end up in senescence, which is bad, and it occurs when you're aging. So it's been shown that diabetic individuals who take metformin have reduced telomere shortening compared to those who are not. And more interestingly, In mothers with diabetes, metformin treatment reduced telomere shortening in their male offspring. The last and my favorite mechanism is the epigenetic one. So epigenetics, by definition, are a heritable change in gene expression that's not caused by changes in the DNA sequence. And the epigenome is made up of chemical compounds and proteins that attach to DNA and modify its function. For example, methylation, histone modification such as methylation acetylation, RNA interference, and long non-coding RNA. This is really fascinating because two individuals with the same exact DNA, not necessarily, they don't express the same genes because their methylation patterns may be different. For example, in twins at a young age, they have the same methylation patterns, but as they grow up and grow up in different environments, they have very different methylation. So methylation can also be used as a biological clock because when we're younger, we have less methylation in our DNA, and as we get older, we have more. However, methylation can also be affected by other things such as stress. If you're stressed all the time, you're going to have more methylation. So as you get older, scientists can look at your DNA, look at the methylation patterns, and pretty much guess how old you are. So for methylation DNA, it's important to note CpG islands. So cytosines are methylated when they are 5' to a guanine. When this happens at a promoter, transcription factors can't bind, which doesn't allow RNA polymerase to bind, which means the whole gene that's controlled by that promoter will not be transcribed. So if there's heavy methylation, that corresponds to less transcription and less expression. It also has another mechanism, MECP2 and HDAC. So if there's a lot of methylation, instead of getting there's no transcription factors. Instead, there's MECP2, which goes there, which attracts HDACs, which are histone deacetylases. So it removes acetyl groups from histones. As you see in the picture in the top right, the histones have tails, N-terminus tails, and there's a lot of amino acids on them. And one of them, there's a lysine, and if there's an acetyl group attached to the lysine, it's neutrally charged. But if the acetyl group is removed by HDAC, histone deacetylase, the lysine becomes positively charged. Since DNA is negatively charged and the histone will be, the lysine at least, is positively charged, they're going to attract and the DNA is going to be more tightly wound and the linker DNA between the two nucleosomes will be shorter. They're going to be closer together, more compact, which hides promoters and does not give access to transcriptional machinery, which means methylation and deacetylation decreases transcription. So metformin affects epigenetics by controlling expression at the transcriptional level, and the human body must control levels of gene expression very precisely or something very bad will happen. There are different pressure points to control, but metformin controls gene expression at the transcriptional level. It does this by two mechanisms, by AMPK-mediated TT phosphorylation. So the activation in TT promotes demethylation. So metformin can demethylate DNA by removing methyl groups. It means if methyl groups are removed, there's going to be more access for transcriptional factors which recruit an RNA polymerase and transcribes the DNA into RNA, which then can be made into protein. However, it can also do the opposite by the SIRT activation in HDACs, which we talked about in the previous slide. So SIRT2 activation and recruiting HDACs. So by recruiting the HACs, acetyl groups are removed, which causes nucleosomes to be closer together, which is basically methylation and decreasing the access for transcriptional machinery. So the first mechanism demethylates and allows portions of the genome to be expressed more, whereas the second mechanism does the opposite. It makes portions of the genome less accessible and decreases transcription. Thanks for listening, and I hope you agree with me that metformin is indeed a miracle drug.